Welcome to the highlight session. We will play 4 fascinating and unpublished research highlights, followed by 2 really interesting open access paper highlights. These 1-minute recordings will be played, and then we will head to the sessions to chat with the authors or their representatives. Please vote for the best research highlight using the poll, and the winner will be announced at MVIF number 3. The first research highlight is presented by Chris Severyn from Stanford University, California, USA. The title is The Impact of Gut Decontamination on the Intestinal Microbiome Composition and the Risk of Bloodstream Infection in Pediatric, Allogeneic, Hematopoietic Stem Cell Transplant Patients.
For children with relapsed leukemias and non-malignant blood disorders, hematopoietic transplantation offers the possibility for cure. Infections and acute graft-versus-host disease are the leading cause of death in this transplant population, and being able to find ways to decrease these risks could dramatically improve survival outcomes. One possibility is using non-absorbable antibiotics to alter the microbial composition of the digestive tract, called gut decontamination. We show the results of the first phase 2 randomized trial in pediatric allogenic transplant patients, investigating how decontamination changes the gut microbiota. We present both clinical outcomes data as well as look at genomic sequence of bacteria causing these bloodstream infections. Using a strain-specific analysis tool developed by Matt and colleagues at the Banfield lab at UC Berkeley, we compare the DNA of any bloodstream infection-causing bacteria to the stool from patients and found that the gut microbiome may be a reservoir for a broad range of infectious organisms. To learn more, please come by and visit our research highlights.
Next research highlight is presented by Djamila Eliby from the University of Melbourne, Australia. The Gut Microbiota in Anxiety and Depression, a Systematic Review.
Today I'm presenting findings from a systematic review of the gut microbiota in anxiety and depression. Included were 26 studies in humans with 21 in clinical cohorts with depression or anxiety and 5 studies assessing symptoms only. Diversity findings were inconsistent, with more studies reporting no differences in alpha diversity between clinical groups and controls, and there was little consensus on beta diversity differences. However, taxonomic findings indicated that both disorders may be characterized by higher abundances of pro-inflammatory taxa such as Enterobacteriaceae and Agathella, and reductions in anti-inflammatory short-chain fatty acid producers such as Faeculobacterium and Coprococcus. The microbiota-mediated inflammatory response may contribute to the pathogenesis of anxiety and depression via communication of peripheral inflammation to the brain. However, the influence of confounding factors such as diet and psychotropics need to be more carefully assessed in future research, along with an essential focus on microbiota function.
The next research highlight is presented by Smitha Sukumar from the University of Sydney, Australia. The composition of the oral resistome is altered by the presence of caries.
Hello. Our study looks at the collective of antimicrobial resistance genes, or the resistome, in the mouth and how it's affected in health and disease. Caries or tooth decay is a multifactorial lifestyle disease associated with dysbiosis of the oral microbiome. However, the notion of bad bacteria has not been borne out by taxonomical studies, which means functional characterization of the oral microbiome is required, including understanding its resistance potential. In our cross-sectional metagenomic study, we found no significant difference between caries-affected and caries-free groups of children in terms of alpha diversity and overall abundance. But differential abundance analysis revealed 5 antimicrobial resistance genes that were drivers in the caries-affected group, and these genes were more likely to be associated with more species compared to the caries-free group. This suggests important associations may exist between the structure of the resistome in caries and health. Thank you.
This next research highlight is presented by Svetlina Vasileva from the Queensland Brain Institute in Brisbane, Australia. Maternal immune activation and the gut microbiome in offspring.
Maternal immune activation is the activation of the maternal immune system during pregnancy and is a well-known risk factor for autism spectrum disorder. MIA has been associated with a disturbed gut microbiome in animal studies, but clinical research is still missing. So here we investigated the relationship between MIA and the gut microbiome of 174 children. Consistent with previous research in ASD, we found that children born to mothers with MIA were more likely to be diagnosed with autism. However, unlike animal studies, we found no significant differences in microbiome richness and microbiome diversity. We found significant difference in only one bacterial species, Thycalicatena torquis, which was significantly more abundant in children of mothers with MIA. Given the current interest in microbiome-focused therapies, we should exercise caution in translating microbiome findings from preclinical models to a clinical setting. Thank you.
The next talk is an open access highlight presented by Aayushi Uberoi from the Department of Dermatology at the University of Pennsylvania, Philadelphia, USA. Commensal microbiota regulates skin barrier function and repair via signaling through the Aryl hydrocarbon receptor.
An intact skin barrier is critical to human health. A damaged skin barrier increases susceptibility to pathogens, inflammation, and dehydration. The skin is home to the skin microbiome, which is critical as it protects against pathogen colonization and is fundamental for training the immune system. We wanted to test the contribution of skin microbiome in regulating barrier repair and integrity. By comparing epithelial transcriptomes of germ-free mice raised in absence of microbes to that of conventional mice or specific pathogen-free mice that have a microbiome, we found that germ-free mouse skin was downregulated in expression of genes involved in epidermal differentiation and barrier function. By performing ultrastructural analysis and barrier repair assays, we found that specific pathogen-free mice had improved skin barrier and repair function. We found that this activity was by regulating the aryl hydrocarbon receptor, which is a xenobiotic sensor that recognizes several environmental signals. Finally, we found that by using a synthetic community of commensal microbes isolated from healthy human skin, we could improve barrier repair function in germ-free mice and models of skin barrier disruption. This activity was dependent on the aryl hydrocarbon receptor signaling pathway. In conclusion, we demonstrate that commensal microbiome are critical regulators of skin barrier function. I invite you to discuss these findings in the virtual room with me. Thank you.
And the final presentation is an open access highlight by Wodan Ling from the Fred Hutchinson Cancer Research Center, Washington, USA. Powerful and robust non-parametric association testing for microbiome data via a zero-inflated quantile approach, ZINQ.
A recurring objective in microbiome data analysis is to identify individual taxa which are associated with diseases or exposures. But this analysis is challenging because microbiome data are usually zero-inflated or dispersed and heterogeneous. So classical tests will lose power, and tailored tests usually cannot control type and error, and most of them cannot detect heterogeneous effects of the clinical variables. So we developed ZINQ, a powerful and robust alternative. First, we do differential tests for the proportion of zeros based on logistic regression. And second, we do differential distribution tests for the non-zero part based on quantile regression. And finally, we combine the p-values to reach the final conclusion. By simulation and real data analysis, we demonstrate that ZINQ can control type and error very well. At the same time, it will increase the testing power by identifying heterogeneous effects of the clinical variable. I look forward to discussing more details with you in the breakout room. Thank you very much.
Thank you so much for joining the MVIF2 event today. We will now head to the sessions for the rest of the conference, and you can ask your live questions to relevant speakers. Most speakers have kindly made themselves available for questions, so please do join them to discuss their work. There will now be a quick video which explains how you can get the most out of the sessions.